thank the organizers. Anyway, so my lab is interesting in basically looking at the imbalance between excitation and inhibition. And we did a lot of work of glutamate receptors, but now I'm going to show you how GABA system, inhibitory system are involved, but mainly in conditional reward, in cocaine conditional reward. This will be the main thing that I'm going to show. Okay, so for this audience, I don't need to say that it's, addiction is a devastating disease, but they're always impressed by this picture. Sorry? Always impressed by...
תוכל להישאר לעמוד ליד הפודיום, בסדר?
לעמוד, אוקיי, כן, אין בעיה. אני רדיואקטיבי קצת, אז זה בעייתי, אבל בסדר. בואו ננסה, נו. הוא לא עובד.
תישאר עם המיקרופון בממה. בממה הוא עובד.
עכשיו? Anyway, so I will scream. So I'm really impressed and always start my talks showing this picture this cartoon, because I think this shows really, not only one example of how this disease is really devastating. And this is the glucose utilization in a normal brain, and look the drop after 10 or 100 days of cocaine use. So the brain function is reduced, and it's not reversible because I'm not seeing it here. There is another study that shows that after 7 years of withdrawal, it still never goes back to baseline. So something happened in your brain, and there is no cure for it, mainly in relapse. And the numbers are growing. This is a World Drug Report that goes every few years, and you see the numbers in million. Note one in any less than 10% goes to treatment. Many reasons, and we spoke about it yesterday and today. Why not? So it's the numbers are growing. No medications. It's a big problem. And my lab again is focused on molecular neurobiology of addiction, looking at the molecules, and I will zoom in into the molecules, and we can split it by this very nice cartoon that Volko and Peter show a long time ago. And the transition to addiction in terms of molecular changes can be split into 3 stages. The acute phase. In the acute phase, you have a lot of changes. This is a protein function. Okay, so any kind of protein function, change in protein function. So acute use, you have a transient change. It fades away very quickly, and it goes back to baseline. And then when you start to be compulsive, frequent user, the magnitude of molecular change is going up in a way more than the acute change, because if in other words, this is many frequent acute uses, but it builds up, and you have this big change, and it fades away, and you assume to think that after withdrawal, whether it's forced withdrawal or by choice, it fades away, and that's it. The brain here should be the same, but no. There is another wave, which is the killer wave, is the wave that comes following withdrawal with no drugs, and it goes up and up over time, meaning that the chances to relapse here is bigger than here. So, and I'm a former smoker, okay, and I quit only by mind with no help from other things. I know, I've been through this thing, okay, and you have a lot of relapses, and so something happened in your brain that you can't just get rid of it, okay, and it becomes bigger and bigger. So after 10 years, having 1 cigarette will take you back straight to your 2 packs a day. Anyway, so the problem is here, and we try to see what's going on here. So the connections, okay, we're all familiar with the connections, and I'm focusing on VTA dopamine neurons and their projection to the accumbens prefrontal cortex and the mutual projection back to the VTA, focusing on the inhibitory system. And as you all know, glutamate is innervating also the VTA, as well as dopamine and GABA, and all 3 together form this modulation. And again, any change in excitation inhibition balance will cause a problem, okay? And just for Yoni, and not to be offended by Yoni, okay, there is a very small neglectable area that Yoni going to talk about. Another source of which I'm not going to talk about is the ventral pallidum, of course, will play a critical role in this modulation, and Yoni will talk about it. Then we're focusing on the contribution of GABA A, the main inhibitory ion channels, to VTA dopamine neurons during conditioning. So the first, and I will briefly just highlight what we did a long time ago, and I will show you what we have now. So Avner asked the basic questions. He did horizontal sections electrophysiology on VTA dopamine neurons, and he asked the basic questions: how cocaine or dopamine, in our case, because it was easier to work with dopamine, affect GABA iPSCs in VTA dopamine neurons following that application. So basically what we show, and this is the main result, the dopamine inhibits GABA A iPSCs in the VTA, which makes sense. Less inhibition, excitable VTA dopamine neurons, which then will release more dopamine to the nucleus accumbens. You get all the euphoria, all the things that you know. We also show that it's D1 dopamine receptor mediated events presynaptically, which then decreases the dopamine release, increases the dopamine release and decreases GABA inhibition. And following exposure, you have this upregulation or hyperexcitability of these dopamine neurons. And later on, and again, I'm not going to elaborate, we did a lot of studies, we have like 6 or 7 papers about this, and showing that under different conditions of GERK, for example, GERK channel inhibition, you see a rebound GABA potentiation. So at the beginning you have inhibition of GABA, in other words, disinhibition of dopamine neurons, and then rebound potentiation, which makes sense when you take cocaine. You have a high. I never tried, so I don't know. It's like just rumors. So you have a high, and then you have a down state, and back to baseline. So it's not always high. The high is terminated at some point. So the question, okay, so and to summarize this, we also show that it's, again, D1 GERK-mediated events. Also, we show that it's NO-mediated events because it was blocked by inhibitors of NO, like L-name, as Miriam showed before, and that was the conclusion. But when you look at VTA and you see the GABAergic inputs, so you have some coming from the RNTG, which were mentioned by Miriam, and some comes from the nucleus accumbens, either directly or through GABAergic interneurons, and some comes from the lateral avenula, which I will talk later on about it, and again, straight to the RNTG, and 15% of this glutamatergic neuron synapse onto GABAergic interneurons. So you have different sources, including ventral pallidum, but again, tend to be ignored, and all are contributing to this inhibitory effect. Now, when you record with electrical stimulation, like we did before, okay, you evoked electrical field, and you stimulate all the GABAergic sources. You don't really know which contributing to what event. And you're recording from the soma for your question, and you have a total event, and you don't discriminate which input is contributing to this total event. In other words, the inhibition that we saw is due to maybe all the inhibitory input, or part of them, or only one of them, and so on. Now, to test this directly, we use channel rhodopsin and optogenetics, and we injected RMTG neuron or nucleus accumbens. We try also with the lateral avenula, but it's still going on, and we have no results. And I will show you so the question was whether its input disinhibition or disinhibition of dopamine neuron is input specific. It's whether it's coming from the RNTG, nucleus accumbens, or both, okay? And what contribution each input has to this inhibitory effect, and of course, what the behavioral consequence. So this is just to show that when we inject channel rhodopsin into RNTG, it specifically goes to the VTA marked by TH, and you will nicely see when you do a high magnification, it synapses onto VTA dopamine neuron. So when we did this, and we optostimulate the VTA with the optic fiber, and you can see very nicely the inward response of iPSCs. This is the brief light stimulation, of course blocked by picrotoxin to show that it's GABA A specific inhibition. And when you add dopamine to it, you see a great inhibition, okay? Way more than we have with the electrical stimulation, almost 90% decrease in GABA response. And this is just to see that not all the cells responded to light, okay, meaning that, and it's segregated, we can't really say that it's in a specific part of the VTA. Now we are trying to increase the number to see whether we see this discrimination in terms of where it's coming from. But you can see that light insensitive neurons, most of them were sensitive, but some of them were not sensitive to this light stimulation, meaning that this input is not only, not generally affect all the neurons in the VTA, and what's the specific role we still don't know. Okay, so we did the same thing with the nucleus accumbens. And this is the injection site of the accumbens, and this is we can see it here with this light. But you see it very nicely in other regions, which can be very interesting, but we don't focus on this. But you have projections to the nucleus accumbens as well, so we are ensured that really the neurons are sending the projection to the nucleus accumbens, and we repeated the same experiment. Surprisingly, we also see a big inhibition here of GABA response following dopamine, again blocked by picrotoxin to show that it's specific. A little bit less than we saw with the RMTG, but both of them are way greater than we see with the electrical stimulation, meaning that you have a specific inhibition which is input specific. And for our surprise, most of the neurons didn't respond to light. So maybe the one that didn't respond to light have different roles. We still are trying to figure out what's going on with this neuron that didn't respond, but the majority didn't respond. But those who do respond, then they have this modulation of these GABAergic iPSCs. Okay, and then we ask, okay, so if this is the case, then maybe we can overcome this inhibition by stimulating RMTG during conditioning to cocaine. So we choose to look at cocaine condition reward. Now we're trying to do it in other types of addiction model, but for now we have this condition place preference, and I'm sure everybody knows what it is. So you condition the animal in one compartment to cocaine, into the other to saline, and then you put them in the middle after conditioning, and you let them freely go to each the cocaine, and they will spend most of the time looking for the cocaine. So what we did, so we injected either RMTG or nucleus accumbens with channel rhodopsin, and we optic stimulated VTA dopamine neurons during conditioning. Through the whole session, it was about 15 minutes of session, through the whole session, with a pulse, I think it was mimicking excitation or LTP. Okay, and we did like 4 consecutive sessions of stimulation, and then we put them in the middle and asked where you want to go. So the control group show very nicely the preference, but the one that got cocaine plus light stimulation show no preference whatsoever. So we abolished the preference. We still don't know how it happens, whether we interfere with the memory, it's still ongoing research on how the nature of this interruption, but you clearly can see that condition is abolished. I mean, the expression of conditioning is abolished. Same with the nucleus accumbens. Again, although most of the neurons didn't respond to light stimulation in the acute phase, but most of the, and the little that is still responding, doing basically the same thing. When you stimulate during conditioning in the nucleus accumbens fibers, you still get the same results. So these 2 targets now can be a target for stimulation to reduce the rewarding properties of cocaine with a mechanism which is still under investigation. The other thing we ask whether FOS is just another proof that we are shutting or interfering with the activity. We look at FOS neurons in both areas, plus or minus stimulation, and you can see the drop in FOS activity compared to the control, the cocaine without the optic stimulation, with all the other controls that I'm not showing here, meaning that enhanced GABAergic input attenuates expression in the VTA and reduce neuronal activity. Okay, the other thing that we measure is the biochemical change, and again, we are looking at machinery and the proteins that underlie this, all these processes, and one of the things that we measure always is the excitation, and it will balance and focusing on glutamate receptor as well, and just a brief description. So this is the AMPA receptor, main receptor of glutamate, and it has a key role in synaptic plasticity and memory. And we previously showed that phosphorylation here play a critical role in maintaining memory. And we show here that phosphorylation can erase memory if you under the, different conditions. I gave this talk 2 years ago. We can erase this memory by a small peptide called ZIP. But the point here that just to show you that when you manipulate this phosphorylation site on GLUA1 AMPA receptor, which is again the key player in long-term potentiation and excitation, and it down-regulates AMPA receptor, and that's how you erase memory. So looking at these components, and I show only some of them, okay, here biochemically. So when you look at ERK, which is responsible to initiate this process, and the phosphorylation of two sites, S serine 845 or 31 on GLUA1 AMPA receptor, you see very nicely that cocaine elevates 3 of them, and cocaine by stimulation reverse completely the biochemical change. And for me, as a molecular person, when you see this evidence, biochemical evidence, it's proved that this is what's happening. I mean, in the molecular level, if you see change in biochemical response to a stimuli, then it's make the evidence stronger that this is really what's going on. You interfere with the cocaine-induced changes, adaptations, if you want, in all the excitation inhibition molecules that are responsible for it. And the key molecules, one of them is AMPA GLUA1. Okay, so we mentioned the medial prefrontal cortex and the critical role in many aspects of addiction. And I'm not going to elaborate why we had this project going on, but the next idea was to try to activate the lateral avenula, which was mentioned here. By the way, again, lateral avenula projects almost 90% of the glutamatergic projection to the RMTG to inhibit VTA, if you want. And this is considered as the anti-reward system, and it mainly responds to punishment, to something bad, okay? So the idea was to take advantage of the connection or projections of the medial prefrontal cortex to the abenula and stimulate the abenula during condition-place preference. So first we verified that indeed, so the protocol was like this. We injected channel rhodopsin into medial prefrontal cortex, optic fiber above the abenula, conditioned the animal same way that I did before with the RMTG, and we made sure that indeed the projections from the abenula and from PFC, now you see only one side, is indeed are mutually connected, and we injected retrobeats to the lateral abenula, and we saw very nicely how the trace is in the medial prefrontal cortex, suggesting that indeed you have this stable connection between these two regions, and when you stimulate again in the box while conditioning the lateral abenula, we abolish completely place preference. Meaning that again, another pathway, another circuit can be involved in the rewarding properties of the drug. Now, this is again not an addiction model, it's a reward model or reward and aversion model, and also seeking model. Now we're trying to see what's going on after withdrawal. And I will show just one example, but just finishing this. This is during acquisition, we don't know the mechanism, but it's very promising that this pathway can interfere with drug seeking. So the next thing that we asked, but just to complete this, I forgot to have the other slide. So the same thing we repeated with the first positive neuron, and you see when you stimulate the abenula, you have an increase in force activity, meaning that you really activate the lateral abenula during this optic stimulation, and it's when you are on cocaine without the stimulation, the lateral abenula fairly is active normally, but when after stimulation you indeed activate this region, to maybe go through the RNTG and disinhibit or inhibits dopamine neuron, like I showed you before. Okay. Again, biochemical evidence to show that it's really indeed working. This is only just the levels of GLUA1, okay, in the synaptic membrane. You see in the prefrontal cortex a reduction in GLUA1, increase in the habenula and the VTA, and in all normalized by optic stimulation. So this intervention really can reverse the molecular adaptation, both electrophysiologically, as you saw, and by this measure of biochemistry. Okay, so now we are doing a bunch of experiments to show, okay, to translate it into the beneficial effect of the stimulation during withdrawal, because this is the main problem, right? We want to treat people that got addiction and now trying to prevent relapse. So, and again, we are focusing on conditional preference. So one of the examples that we doing now, and this is not published yet, is to see after conditioning with no optic stimulation, what happened during withdrawal after conditioning and drug seeking when you reinstate the animal with cocaine. But this raises a lot of questions, but what we're going to do here, and this is just preliminary result, is to see whether if we stimulate or condition the animal with the optic stimulation in their home cage, after conditioning in the regular apparatus, and we put them back in the apparatus, if they will show condition place preference. Meaning if we can transfer the context and give the treatment somewhere else, whether they will abolish this expression in the original context, as you can see here, otherwise I won't show it to you, that we indeed can do this. After stimulation, in a different context, the transfer of the context or the de-evaluation of the reward can happen anywhere. In other words, if you want to translate it into rehabilitation, you can have this treatment somewhere else, not necessarily at the same place where you use the drug. Okay, so with this I will finish and thank the people. All of them left the lab already. Now we have a lot of other projects that we're doing that maybe next iCare meeting I will describe, and thank you very much.